Here is a combined summary of the 2 pages. This research presents a spiking neural network (SNN) model that incorporates learnable synaptic delays using dilated convolution with learnable spacings (DCLS). The model is trained on the Raw Heidelberg Digits keyword spotting benchmark using backpropagation through time with surrogate gradients. Analysis of the spatio-temporal structure of synaptic interactions in the network reveals that excitation and inhibition are grouped together both in space and time after training. To enhance the efficiency and biological realism of the model, a dynamic pruning strategy is implemented, combining deep R for connection removal and RigL for connection reintroduction. This ensures the network maintains optimal connectivity throughout training. Additionally, Dale's principle is incorporated, enforcing each neuron to be exclusively excitatory or inhibitory, aligning the model closer to biological neural networks. The results show that the spatio-temporal patterns of excitation and inhibition emerge in the more biologically plausible model as well. The research demonstrates the potential of integrating learnable delays, dynamic pruning, and biological constraints to develop efficient SNN models for temporal data processing. SNNs are inspired by the functioning of biological neurons, processing information through sparse binary spikes, enabling efficient processing of temporal information. Learnable synaptic delays, which can vary across connections, allow neurons to perform coincidence detections across longer time intervals, enhancing SNN's ability to process temporal information. The use of dynamic pruning and rewiring techniques, such as deep R and RigL, aims to emulate the biological efficiency of neural networks, potentially offering superior sparsity and accuracy with fewer floating point operations. The analysis of the learnt parameters of the network is conducted, and the same analysis is performed on the more biologically plausible network, enforcing biological constraints during training. The leaky integrate and fire (LEAF) neuron model is used, with membrane potential dynamics governed by equation 1, and spike trains generated according to equation 2. The research enhances the understanding of spatio-temporal dynamics in SNNs, suggesting that the spatio-temporal features which emerge from training are robust to both pruning and rewiring processes, providing a solid foundation for future work in neuromorphic computing applications. The research paper discusses the structure of excitation and inhibition in spiking neural networks, SNNs, by Mészáros et al. It begins by describing the discrete update equation for the membrane potential, which is numerically integrated using a linear Euler method. The Heaviside step function is replaced with an arctan surrogate gradient for training. The synaptic delay training is implemented using dilated convolutions with learnable spacings, DCLS. For dynamic pruning, the authors combined deep R for dropping connections and RigL for introducing them. The deep R method defines synaptic weights based on a constant sign value and trained parameters, encouraging pruning of unnecessary connections. RigL introduces synapses based on gradient analysis. The paper also discusses the assumption of adhering to Dale's principle, where neurons are either exclusively excitatory or inhibitory. The authors extended the PyTorch implementation for their experiments, including a hidden layer with 256 neurons, dropout layers, batch normalization, and delays. They trained the model with the Atom Optimizer and one-cycle scheduler, achieving 94.5% test accuracy on the Raw Heidelberg Digits dataset. The paper concludes by analyzing the learnt spatio-temporal patterns through spatio-temporal receptive fields. The research focuses on the structure of excitation and inhibition in spiking neural networks, SNNs. It explores the emergence of spatio-temporal patterns in both unconstrained and biologically constrained networks. In the unconstrained network, a clear pattern of excitation and inhibition is observed after training, as illustrated in Figure 2. The 2D autocorrelation analysis using Moran's I indicates a moderate level of spatio-temporal autocorrelation across all output neurons, with the highest value observed for class 14. This suggests that even in the absence of biological constraints, coherent patterns emerge. In contrast, the biologically constrained network, depicted in Figure 3, shows a weaker emergence of spatio-temporal patterns, with an average Moran's I value of 0.0184. This is expected due to the sparse connections within the 8 neighborhood. The study also compares the classification performance of dense and sparse Dalian networks, with the dense network performing better. This research provides insights into the dynamics of excitation and inhibition in SNNs and highlights the importance of biological constraints in shaping network behavior. The research focuses on the structure of excitation and inhibition in spiking neural networks, SNNs, and their efficiency in machine learning tasks. It explores the effects of structure learning and delay learning on classification accuracy. The authors compared dense and sparse network architectures, highlighting the benefits of sparse connectivity, especially with Dale's principle. They also analyzed the combined and separate usefulness of dynamic pruning and delay learning, concluding that dynamic pruning is vital for maintaining high performance in sparse networks. Delay learning, on the other hand, seems beneficial when the structure is fixed but less clear when the structure is learned. The study emphasizes the importance of structure learning over delay learning and suggests that a good baseline for delay learning methods is testing their performance in sparse settings. Overall, the research aims to pave the way for future studies into optimizing SNNs for various applications, particularly those with memory and computational constraints. The research by Mészáros et al. Focuses on the structure of excitation and inhibition in spiking neural networks, SNNs. The authors declare no conflict of interest and acknowledge the support of a doctoral scholarship by the Leverhulme Trust and the EPSRC. The code and generated example networks will be published upon manuscript acceptance. The paper references various studies on SNNs, including works on error backpropagation, learning delays, and neuromorphic hardware systems. It highlights the importance of understanding the balance between excitation and inhibition in SNNs for efficient computation and learning. The authors also discuss the role of polychronization and dilated convolutions in enhancing the performance of SNNs. The summary covers key concepts, methodologies, and significant findings from both pages, providing a comprehensive overview of the research.